Thanks, Nancy, and thanks for inviting me again. You seem to be maybe even becoming addicted to the torture of people like us coming up here and talking with you today. I want to talk a little bit about space cell biology and some of the smaller things that are living and some of the things that go on with that could be useful to us and from understanding those basic biological mechanisms. But it does give us a window into something that when I was taking biology as an undergraduate and even as a graduate student, no one talked physics, and there was no cell biology book that had an equation in it that had G in it, G for gravity. So we've started, to come along a pathway now where I think this is what's going to happen. We're going to see and try to understand much more about physical forces and their effects on life. You've all been introduced to this shopping list of, risks to humans in space. I don't think I need to go over it. I hope I don't. But I will not talk about any one of these specifically in any great detail. But instead, we're going to take a look and see if we can get our mindset to start looking at ways that cells and bacteria, basically unicellular life, can help with regard to our exploration of space and actually going to Mars. These are convenient models for higher life forms. It's well established. We study cells. A good 35 to 40% of the research that's funded by NIH is actually cell-based research. And so you can see how important it is to have these as models. They require simpler accommodations. We have to satisfy all the needs of the crew and all the people that are involved inside the habitats and systems like that. The cells don't really care. And the other thing is they don't have attorneys and they don't make law, they don't bring lawsuits. So we can get a lot of information by studying cells under these. And first off, and secondly, it is, it's acceptable for us to take cells and bacteria into these really hazardous environments and begin to understand how the mechanisms work. And when I say it's an acceptable risk, for humans we have a lot of clearances and we have a lot of things that we have to do and understand about that human and their physiology before we can park them in one of these hazardous environments. So it has a shorter generation time. Bacteria about a half an hour or so. And you say, well, who really cares? But if you're looking at the effects of space on a living system, and you want to know from generation to generation, are we building something into that generation that we didn't want? Okay? Something that could be hazardous to them, something that could be untoward in some way or another. Well, with that kind of a generation time, you get 5,000 generations on a 100-day increment on the space station. 5,000 generations. With human cells, we get 100 generations. With human beings, we get about a tenth of a generation. So you can see what I'm saying here. The comparison is a lot of information can be obtained. They can give us preliminary answers to a lot of questions. And the other thing about it is when you're working with cells, and I think you've been introduced to this term analog. An analog is a condition that ostensibly gives you some of the same results that you would get in microgravity. And we have a number of different kind of analog systems for cells. And so you can do a lot on the ground as a prequel, so to speak, to going to space and finding something out. So that's basically the kind of direction that we want to go today. I'm not going to give you an avalanche of data, but we're going to actually have a dialogue on some concepts. But these are the kind of questions, how do changes in individual cells go, scale up through the tissues in the organism. We're not the only ones asking that question. We're going to ask it in the context of space. It's asked every day at NIH and NSF and research institutes all around the country. Can we use cells to conduct missions with unknown consequences? The answer is yes. With unknown consequences, it's very difficult for us to take people someplace. But we can put cells into anything we want, and we can collect the information and get answers that we need, and we're not at risk for something that would be considered unethical. Can we use cells as sentinels? They used to use canaries in the coal mines years ago. You're all probably too young to even know anything about those. They're probably on any coal mines around you. But they were there because of their exquisite sensitivity to carbon monoxide. And so the cage with the canary, when the canary keeled over, it was time to get out, right? Because they had a much greater sensitivity and they would die long before a human being would. So can we use them as sentinels? And that I think would be a crafty use. And the only thing we have to be concerned about is protecting the planet. And that's both of them. And even the other planets. We have planetary protection programs inside NASA that are really stringent on what you can take and what you can use and what you can leave on the surface of the moon and surface of Mars. So these are the reasons. That's the kind of justification. This is something that, if you haven't had it yet, you really need to see and hear. Okay? Life evolved on this planet over the last 4.8 billion years. And a lot of things changed. There were a lot of different things, chemical compositions, even the physical forces at play in hydrodynamic shear and other things like that had great variability over all that period of time. But the fascinating thing about it is we've been at 9.8 meters per second squared acceleration. That's the force of gravity, okay, for the entire time. So the history of life on this planet, as we understand it and know it right now, has never had to deal with a diminution in that force. There are accelerations that can make the force greater, but not on the lower side. The lower side is something that's reserved for being in space. And so that exception makes this area of research incredibly stimulating, very interesting, and probably outstandingly important because, yeah, it'll get us to where we want to go. But it's also going to tell us what gravity did, okay, in shaping life the way it is here on the planet. And when you look at these forces that are in nature, basically, I've got it, this is very rolled up as far as the four categories, but we have gravitational forces, which if you're listening to the news, we're looking at waves, gravitational waves now. But we also have electromagnetic forces. We have intermolecular forces that are either strong or weak. Strong ones like covalent bonds, weak ones like van der Waals, dipole-dipole interactions, hydrogen bonding, and those kinds of things. The interesting thing about this is when you rank these, gravity is actually the weakest force. It's the weakest. You can oppose gravity very easily. We do it. Get in an airplane and we go. Put it in a rocket, take a ball and throw it up in the air. You can oppose it very, very easily. If I take a piece of paper and tear it, how many covalent bonds do I break? That was on the test. Okay, it's done, right? Yeah, the energy of activation and the energy of hydrolysis of a covalent bond is much, much higher than you can get by just merely tearing it. So you can see what we're dealing with when we talk about gravity. It has such a huge sphere of effect. The intermolecular forces require close proximity, but in that first picture you looked at up there, it's actually a galaxy similar to ours. We can't take a picture of our own galaxy like that until we get out of here, and that's going to be a long time. But you can see that galaxy has a tremendous amount of interaction and organization. Much of that is due to gravity. So despite the fact that gravity is the weakest force, it has a huge sphere of influence, comparatively speaking. So those are kind of introductions to that. This would be really nice if I could get to, oh, I see what I did. I shouldn't complain. It's on video when I do this. So I already did this. I told you where we are with the gravity as being the weakest one. And, how do we study these influences? I talked about microgravity analogs. And we can also do more computational modeling now. And there's a lot more mathematical basis to what we understand structurally and operationally that goes on inside cells, inside human beings, and even inside populations. And so that's going to be extremely useful to us. And you'd say, well, I don't think you can replace this with a computer. No, you can't. But what we can do is that we can actually build much better experiments for space if indeed we model them mathematically and do the experiments, as they say in silico, at the workstation, and I can do 1,000, 10,000 experiments in a month, all in computational model, and then take 2 to 5 of them to the bench instead of spending many, many years and millions of dollars. So that's where we're headed. So what's the basis of the cellular response to microgravity? The theory was, originally, when I first got to NASA, the theory was that cells were too insignificant in mass to be affected by the loss of gravity. And if you look at Einstein's equation, E equals mc squared, m is mass, and the mass of a bacterium is vanishingly small. And so it seemed insignificant, and it shouldn't be affected. But the truth is, they were. And as we move along further and further, we find out that they indeed are affected by microgravity, and they indeed respond to it. And we'll talk about those in a little bit. It's probably not going to be exactly the same for both mammalian-type cells and, say, cells at the cell wall, like bacteria as a model cell for that. And so we're very interested in finding out how it is that the changes in gene expression that you've already heard about, whether they're in bacteria in space or they're in mammalian cells in space, how those take place, how those come about. Structurally, those cells are vastly different. And so we're going to get informed in a way that we had never anticipated by taking things and going to space, and not only doing space exploration, but trying to understand life in space. Okay, and the other thing about it is that, you're always concerned that when you have effects being stressed upon cells, things are taking place and they're being stimulated in some way or another, that when you have multiple stimuli, do they have canceling effects? Do they have synergistic effects or whatever? So we don't know if microgravity makes our susceptibility to radiation, for instance. Does it make it greater or lesser? And those are the kinds of studies that you can do in model systems like in cells. So some of the specific questions that will come about as we move into a phase where we're going to be doing space exploration on a regular basis, we're going to go ahead and have established habitats and places to be on Moon and Mars, and we may even have colonization of those places occurring where people will be spending most of their time or even the rest of their life in that space. So you have to understand the adaptive responses. And adaptive responses are troublesome to us in a number of ways. We know a few of them. But the good thing about the adaptive response is when you come back to Earth, you take away the stimulus of microgravity, then we return to our normal ground state, if you will. The phenotypic and genotypic changes that are induced by microgravity become critically important as we understand that, at least for short durations, the times that we've gone 6 months and one person for a year now, it appears that many of these responses are phenotypic. However, as we stay there longer and longer and experience other stimuli, for instance, once again, radiation also known for being mutagenic, that, what happens? Can we have something happen as far as genotypic changes that occur when you come back, they're there, and they're not going away? We'd have to deal with them in this environment or send you back up there. So it's an interesting question. You could make all kinds of cases about it. I think one of the things, I intimated earlier when talking about the constancy of the gravitational vector over the many, many, many years of the development of this planet, that is itself microgravity. Will it be a selective pressure? And if it is a selective pressure, that could mean that you've got mutagenesis occurring, and you could have selections of variants. You could even have something that we might even eventually be able to call speciation occurring. So this could occur when you have organisms with a short generation time or cell populations with a short generation time. When you look at cardiac muscle, I'm not going to be so concerned, about that particular aspect. There are other things to be concerned about. But when you look at gastrointestinal epithelium on a Mars trip, You can have 1,000 replications. It's going to be 1,000 replications that are going to occur in that environment. It will be a low gravity, and it will also have a substantial flux of radiation. It's going to have to be dealt with. So these are the kinds of applications that we can put to work by using these small life systems. Now, one of the major questions that comes up is every time we look at what microgravity does to bone loss or whatever it is, muscle deconditioning, we're going to list a whole bunch of things. I always ask the question, well, how much gravity do you really need? All right? How much is going to be enough to maintain a person? Because if we knew how much gravity there was, maybe we could use, a temporary artificial gravity type system to get people ginned up and get them back to where they're supposed to be. So when you look at the relationship, particularly the relationship between the biological response and gravity, and you can see along the abscissa that gravity is there, that we have data. We have data at between 10 to the minus 5 and 10 to the minus 6, and we've got data at 10 to the 0, which is Earth. We've got data in both places. Big piles on Earth and a smaller pile of data that's in space. And so the question is, which line do you draw through that? Which one is going to be the one that's going to be the relationship? Because we've not done the experiment. This experiment needs to be done. It hasn't been done yet. In all the years that we've been up there doing all the things that we do, we still haven't done this experiment. And it's a critically important question, not only for human health, but it's a critically important question for the design of the habitats, the design of spacesuits, a whole bunch of things that are impacted by knowing just how much gravity is it you need to keep a person normal or close to normal. And so when you look at this, a power curve and linear relationship and an exponential sigmoid curves there might be, which do you think it is? What would be the logical thing? The clue in this question is on the ordinate. When you look at the ordinate, that should tell you something. What do you know about biological responses? The greater predominance of biological responses are in a sigmoid relationship with whatever the stimulus is that you're using. So it's going to be a sigmoid curve. And so for a long time I had the yellow line on there, and the engineers loved it. They'd say, oh man, that means about, maybe at a 1,000th G, that's all they need. 10 to the minus 3, you get about half the effect that you do. And it took me a while before I figured it out, because I went back to some old data that we were looking at on something that had nothing to do with this question. But just looking at the dose response of a specific receptor to a ligand that's binding to it on the surface of the cell. And it's a sigmoid response. And they all occur within 1 to 1.5 logs of normal. That's where they occur. The entire S proceeds through that distance along that abscissa, which means that already by the time we're at the moon, because the moon would be, I don't know how well you can see this, but he's somewhere, got the wrong one, somewhere in this range right here, okay? That's where the moon, because it's 1,6 G. You go up a little bit, right there, and look at the line extrapolated up, and that's what it's going to be on Mars. So it looks like our threshold is in a really tough place. We're going to have a lot of therapy, if you will, with added, applied G to make this work for, say, if you want to do it that way. So we need to see that. We need to be sure that we're correct. Space has fooled us so many times. We do these things and we draw these equations. You notice everything that I had here and I talked about is not data-driven, it's deductive. That's it. And so we have to go prove it. And if it is indeed following the green line, it's a new day for the space program. So anyway, when you look at cells in space, like animal cells, you take them into space, launch them on a vehicle, whatever it is. It can be your favorite vehicle. But interestingly, the first thing that happens is that the cell loses its characteristic shape and becomes more round. So it decreases its surface area to volume ratio, exactly what it's doing. And so in that particular phase, then a whole bunch of things happen. These are lists of changes that occur. And you can pick your favorite ones you'd like to talk about, but obviously things like differentiation, glycosylation, locomotion, and signal transduction are critically important in embryogenesis. In looking at this and understanding this, I'd say right now with our current state of knowledge, procreation in space is totally contraindicated because you have no idea what you're going to come out with, once you fertilize an egg and let it go through all the processes that it's got to go through. So it's interesting in that sense. This raises another question, and we'll come back to this roundness thing here because there's an interesting question that evolves that we'll get to in a few minutes. And these are bacteria, and the bacteria, you don't see anything. You don't see any shape change. You don't see anything like that because it's got a rigid cell wall. Okay, so there are going to be forces against that wall anyway, by the virtue of the fact that the cell inside is different than it is outside. And just like the mammalian cell went round, this one here is probably going to have some pressure differences across that membrane. And could that actually induce the list of things that's going on? Is it possible? Yeah, anything's possible, right? In biology, we say, yeah, it's possible. It's got a lot of probability to it too, because bacteria have structures on their surface that are very sensitive to those kinds of changes, and we'll talk about those in a little bit too. So, and I'm going to leave this. I just essentially said that. We'll leave that alone. There's been a lot of talk in here already about gene expression, so I'm not going to do the gene expression charts and talk to you about it in detail, showing you a lot of data. But we're very early in these studies, and the thing that I want you to know and understand and take with you, no matter what you're doing, whether you're doing strictly clinical work, you're going to be doing some research. Most of what we talk about in genetics in space is a snapshot in time. So you take the stuff up there, and then at some point in time, you hit them with the RNA later as a fixative, you bring them back, and you look and see what the gene expression changes was. You got a picture of what the gene expression's like in that time. That's it. We don't know. Anything else, we don't know what happens before or afterwards. So it needs extensive serial study before you can actually come up with it. And the expectations that this brings is that we're going to get a delineation of the changes that are essential for adaptation. You remember we talked about adaptation, okay, earlier, and we're going to see a much better picture of what genes are absolutely essential to the cell adapting, surviving, and thriving in that environment. And that takes, again, a temporal study. You cannot do that with snapshots. There are also identification of incidental changes. We still have changes that occur in cell, in gene expression. We don't know what the heck they're for. We know what they are, and we know what they encode for, but we don't know exactly why they occur. They are incidental to some of the things that occur. But the ones that I think that we really need to understand are the ones that are actually consequential to the initial changes that took place right at the beginning. A good example is HFR in Salmonella typhimurium. That is a pan-regulating gene. We now know that microgravity changes that gene first. And then following that, a number of other different functions change, one of which is the virulence of that organism, and there's another lecture that will cover that. So you can see how we have to be much more ordered, much more deliberate in what we're asking, and looking for not data as much as we are answers. And Francis Crick stood over here down the hall a number of years ago. That's of Watson and Crick, the DNA fame, structure fame. So, yeah. Stood down the hall here and he said, we spend far too much time in science publishing data and far too little publishing answers. And because at that time, we got into the phase of where we were measuring our progress on how many papers we published and how much data we got. But the fact of the matter is, how many answers did we get, he felt was insufficient. And I think this is going back to that. Okay, now, when you take living systems to microgravity, things can happen. And these things that can happen, we'd like to say and assign them to microgravity did that. That's the cell responding to microgravity. I'm going to make a case for you that those are precious few that actually fit in that category. The indirect effects are due to the loss of hydrostatic pressure gradient. Okay, it's gone. Now, so many things are governed in aqueous living systems here on this planet by the hydrostatic pressure gradient. And you all know what it is, and you know what it feels like when it's, when your ears are not cooperating and you're jiving. So you can get a sense of how profound it can be and how sensitive a lot of living systems are to that. And so no sedimentation occurs. So when you look rheologically at a pond or something or what's going on, there's a sedimentation process that goes on, and a number of organisms deposit themselves on the floor, and then it creates a new biome and all that. No sedimentation. Anything that is particulate, which living cells are, bacteria are, in space, are randomly distributed at the beginning when you first put them in space, inside the fluid. They will change. You don't have any density-driven convection. That's a fancy term for the cold air is coming in from the top, and we feel good. We heat it up, send it back up out of here, and it's cooled off again, and just keeps that cycle going. Well, that cycle doesn't occur in space. And same density-driven convection actually mixes fluids while they're sitting here on the table. You've got your soft drink is not actually still. It's actually got convective gradients going on it all the time. So there are a lot of things that change. Cells, particulates don't interact with the container surfaces like they do here on the ground. Even bacteria will sediment to the bottom of the flask. Okay? And that doesn't occur in space. And so you look at this and you say, I got this response. Right? It's adapted to, and it's now gone, this thing, the cells have gone to secondary metabolism. They're doing gluconeogenesis instead of glycolysis. Well, that's a response to microgravity. And you'd think it might be. I'm not convinced it is at all. Because when you take away the density-driven convection, the only convective forces that we know of that could be of any significance are what are called Marangoni forces. Those are surface tension-driven convections. You can't see them here on the ground because gravity supersedes them and it obfuscates them so you can't see it. But when you're looking at what the response was, you say, well, that's the response to microgravity. No, it's a response to being fed by Marangoni convection, okay, instead of density-driven convection. So you see what I mean by indirect? And we got to be really aware, because there's an advantage to not only saying, I now know what that is. You can do it on the ground, okay? Except for Marangoni, you can do a lot of this hydrostatic pressure gradient loss. These experiments don't require space in their entirety, only for confirmation. They could do, and a lot of this would cheapen the research that we do if we have a better understanding of it. So, let me see the direct effects. Direct, I didn't say it, but direct talks about the potential for a gravimeter inside the cell. Essentially, it's a molecular-based gravimeter or a structural-based gravimeter. Okay, well, I have this sensing these physical changes, that way that might be done and done directly. There is a phenomenon, a theory called tensegrity, advanced by Don Ingber. And it's a very scholarly piece of work that's a collection of papers and reviews that he's written in regards to this. And it's absolutely fascinating. There's also dynamic reciprocity. Mina Bissell, who's famous for the work she's done on breast cancer, looking at dynamic reciprocity. And I'll give you a little pictorial on each of these. And then mechanotransducing channels. And at first, we knew these in bacteria, but we now know that mechanosensitive channels exist along the entire phylogenetic scale. And they're not very dissimilar from bacteria all the way to humans. And in humans, they're rich in a number of different places. One would be the cardiovascular endothelium, but there are a lot of other places. The last one is a weird bird. Okay? It's called the primary cilium. And the primary cilium is a very unique structure that you only have one copy per cell for the entire life of the cell. And some of them keep them, some of them don't. And the primary cilium is a very exquisite force transducer we'll talk about. I'm going to go to the diagrams here. Rather than I want you to take a look at these. In the places where things are converging in a diagram to a point, those could be imagined as focal adhesion complexes. Those are complexes that we find on the surface of the cell that are contiguous with what these structural elements are here of microtubules and and cytoskeletal elements that make up the whole structure. And you can see that there's a theorized organization that's on the left-hand panel there. And when you look at that, you can see that there are attachment points to what is a schematized matrix on the bottom. So that there is a communication that goes on in two ways between the attachments and the focal adhesion complexes through the elastic components that are there for what would be considered a null state, which has looked like this. But here, with acceleration of gravity, you get more attachment points, but also you get changes that occur. And so these spatially are no longer the same as they were before. All right, what happens when you do that is the likelihood is, in signal transduction, we know, whether it's through a focal adhesion complex or not, that there's a lot of proximity that's necessary between enzyme and substrate for those things to actually occur and get the signal across the membrane, down through the cytoplasm and into the nucleus. And so that's what I was intimating when I said to you, when you look at that round cell that we were looking at before, you look at that cell and you say, okay, it became round. Now it is actually a stimulus that the cell is going to respond to. So is it really responding to microgravity, or is it responding to a forced shape change? If it's the former, we got a lot of research to do. If it's the latter, we can do a whole bunch of it here on the ground. I can make cells around on the ground here, it's not a problem. So we could actually look at that entire system, both computationally, because there are models now, but in addition to that, physiologically, we can make the measurements that would give us a good sign as to how things were portrayed out on there. So that's the tensegrity model, which I find very exciting, because when you see this and imagine a cell as this structure like Don Ingber has described here, if you push on one side of it, it's going to displace cytoskeletal elements throughout the cell. Not every one of them, but in that direction. And it'll be directional. Okay? And a cell probably can perceive direction that way too. But at the same time, it can also perceive a change in force. Now, whether the force is microgravity or not, that the tensegrity is responding to, is another question. Dynamic reciprocity. This is an interesting thing. We're looking here at structures within the cell that respond to the matrix, as you were seeing before. But what the tensegrity model doesn't tell you is that when the cell responds to the matrix, which it will during tissue morphogenesis, it's very clear. Until you have matrix, you will not have certain functions occur in the cell. And it gets a signal from the synthesized and laid down matrix that's there. And then that goes back into the cell. But the interesting thing is that as that process goes on, it induces the cell to make other interactions with the matrix. So there's where the reciprocity comes in. They're both giving instructions. Both the matrix gives instruction and the cell gives instruction. So this particular model is going to be very interesting as we try to scale out of cells and into tissues and look at what happens in space, because I can guarantee you that not all tissues in space are going to behave like the cells that make up those tissues when we study the cells in space alone. And so it gives us an idea here that we're probably facing a time when we're going to have to be a much more systems approached in our studies, even at the cellular level. So mechanosensitive channels, I gave you a short intimation of those. And obviously, these channels, I don't know, they really are true force transducers. They're truly a force sensor. So they could be a primary gravasensing type of structure or the system inside the on the cell or inside the cell. And you're looking at two of them here, two views of it. The left one is a view from the top looking down through the channel. And these channels, most of them are ion channels, even when we'll talk about some other ones here in a minute. But most of these are ion channels. And you can see the way that it's structured, that it will take both force that's pushed down against the cell, pushed out from the inside of the cell, but it also, the structures are such, if you look on the left-hand side, you can see, the right-hand side. If you look on the right-hand side, you can see there that there are structures within there that actually can perceive lateral stimulation in the way of force changes. And so you can now see that despite the fact that I told you a lot of the things that we're looking at are effects in the environment caused by microgravity. But I think that there are also things here on a smaller scale and a smaller number that will be actual force transducers that ultimately will become the gravasensor. And the primary cilium is a strange cilium because we normally think of cilia, in the alimentary canal and its role for moving things along. And we think of it in paramecia as locomoting and going around. The cilium that's a primary cilium is non-locomotory. It's if you had it in cell biology, you know that there are 9 pairs of microtubules around the shaft of the inside of the shaft cilium, and there's one central pair. Well, a primary cilium is what we call a 9-0. It's got no central. And if you look at this, you can you look at see if I can get the pointer on it. Here we go. You look in this configuration here, you can see that either one of those, that there are 9 structures all the way around. If you have the central structure, they can whip around. Without the central structure, they cannot. But without the central structure, they are an incredibly sensitive force transducer. And I'll give you an idea as to how sensitive. 10 to the minus, 15, yeah. They're in the femtomolar range, femtonewton range, and that's very, very subtle forces. The mechanosensitive channels are in the piconewton range. And again, we're still in a very, very sensitive range. So it doesn't take a whole lot. There's a lot of subtlety to this, and people will say that, well, there are all these other changes going on. The microgravity change is too small to actually be a contributor to this. And I think we're in the early phases right now, and I wouldn't argue it. I say we need to just prove it one way or the other. And that's basically, I think, the way it should go. But if you don't have this structure, this particular structure, it can mean a lot of problems. If you have it defective, it's a lot of problems. There's actually a clinical syndrome of which there's probably only maybe a dozen in the world with Bardet or Bardet-Biedl syndrome. And this structure is proposed to be extremely important during embryogenesis, especially neurogenesis and the deployment of nerves. It's also very important in the deployment of the organs in the body. And a lot of the asymmetry in organ deployment is due to the fact that there are exquisite responses induced through these particular structures. If you have a defective structure, there are defects in some of the things that I was just talking about. The other thing, like in Bardet-Biedl, they also have very unusual fat deposition. these kids. I mean, it's not typical of anything else that we've ever seen. And so it's got a lot to say about what goes on during embryogenesis. And before we talked about it being contraindicated in space, if we could solve all the other ones that I was talking about before, we'd still have to deal with this one. Okay, this one is a gravasensor in the cell. And I don't know yet how we would do. He would actually do that. Which brings us to a lot of ethical things that we probably need to discuss. And, there are a lot of things that we may have to do ultimately to have people be able to go on these long-term missions. I don't know if you've done the math or sat around or have any idea, but, I get kids saying to me, they go, hey, why can't we be like Star Trek or Star Wars? Why aren't we going, we need to go out there. Why aren't we going to another star? Go someplace else. To them, the solar system is rather boring to them. And so we did it. We just did it on the blackboard. I just went through it with them. These were an advanced biology class in high school. And I said, nearest star, Alpha Centauri, is 4.1 billion light years away. And what's the speed of light? And blah, go through all this, calculate, how far it is in a light year, multiply times 4.1. What's our speed that we can go? Best speed we can do now is about 25,000 miles an hour. With some new ion propulsion that's coming out, probably be closer to 50 to 60,000 miles an hour. But with our current speed, it'd take 106,000 years to get to Alpha Centauri. And so therein lies a real problem. Yes? The guy in Europe who's, I've forgotten his name too, but I think he's Dutch, if I'm not mistaken. Yeah, she's asking about the group that's doing, signing up people to go to Mars. Okay?
Also,
yeah, that's the 100-year Starship. That's what it's called. Okay? But again, once again, propulsion is what's going to determine it. Okay? I have a section here. You can go ahead and look through this called Enigmas. Do you know what a tardigrade is? They're finally out in space now. We've got them out there and studying them because this organism is so fascinating. You can desiccate it down into the Molitor range and you can rehydrate it 100 years later and he's back, reproducing and doing everything else that they like to do. They're only about a little less than a millimeter long. They're 5,000 times more resistant to radiation than human beings. And they've got all of these characteristics that are just absolutely fascinating to me. And it just makes me ask one question. Where'd they come from? We never had that kind of selective pressure on this planet. We've never had that kind of radiation level. We've never been desiccated to that point. So where'd they come from? And so how do you get something like this? And the same is true for Deinococcus radiodurans. This is an organism that is so incredibly resistant to radiation. And so I'll ask a question, two questions here. One is, where did this one come from? It's even more resistant than a tardigrade. But how did it get here? And the second question is, how do you get that level of resistance to radiation? I mean, DNA is DNA is DNA. There's no such thing as bulletproof DNA. Nobody's making bulletproof DNA. So how is it that they can be so resistant? And the way is they fix double strand breaks at such a rate you can't believe. And it's not just one single enzyme, but there's a battery of enzymes that they inherit. And again, we have no understanding as to why. So that's pretty much where I want to go. So I think there's little doubt that cells respond to decreased gravity environments. You notice that it says environments there. The mechanism of gravity-induced responses in cells is still really unclear. But nevertheless, I think it's a great tool. We've studied cells by perturbing them for decades, right? Run water across them at a certain speed, and they respond. That's hydrodynamic shear. They have a full complement of genes to actually survive, thrive, and go on, in a higher shear environment. So our plan is to go ahead and use this as a tool, the cell approach and the microgravity environment. And I think we're going to inform ourselves real well for exploration, but at the same time, we're also going to inform ourselves fairly well for new things that'll come out of biology that we've found, because we didn't know about what's called the heat shock proteins until we heated the cells. Once we heated the cells, we saw the response, the proteins that were produced. We identified the genes, and that's how we got that stress response suite. So I think there'll be the same thing with microgravity. We'll probably have the same. So I'm going to stop there. Got some time? Get a question or two while we can try to do that. And again, thank you so much for inviting me back. Yes, we got one way, way back. So you're going to have to talk real loud.
So I always kind of, I've heard like the micrograph, and I always kind of seen that they were like microscopic, but are they actual visual, like they're visual?
Tardigrades?
Yeah, tardigrades?
Yeah, tardigrades, if your eyes are as old as mine, no. It's not naked eye. You got to have these, and you got, or even a pair of loops, but you can see them, yeah. And if you think they're not around, all you need to do is go out to your yard, pull the grass away a little bit, that mat that's down there, take the mat out, throw it in a jar of water, wait a couple hours, and go back and you'll find them. They're ubiquitous. They're on the Arctic ice shelf. So I have, they're around. And yes, sir.
So people were studying stress responsive cells here on Earth. They're beginning to emphasize the importance of
His question was, seeing how we're looking at the responses here on Earth, the cells are in a 3D matrix. How is that going to translate when we get to space? And very interesting thing, we've done some tissue morphogenesis in space. And the scaffolding that we used to make those constructs on the ground was actually a disc-shaped scaffold. We take it to space, it comes back, and it's actually perfectly round. It's a ball. So they have a tendency to become more round even as a tissue. And that's a little frightening too when you think in the context of embryogenesis. And so we don't know to what extent that is, but we know that a number of tissues do that. All right, let me think. Yes. Asking the likelihood is that we're going to bring a bad guy back from space, whether it's a microbe or a virus or something like that. And what are the chances of that? You can't negate it, okay? Because we actually know, even in our outer reaches of our atmosphere, there are organisms in that environment, in the outer reaches of our atmosphere. And so it's, so far anything that we tested in the way of soil samples from the moon came back negative. Okay, we haven't done enough with the Mars, and we haven't had a Mars sample return so we can actually do it here, so we don't know. But it's going to remain a risk until we can confirm that indeed the rest of the solar system is essentially, as far as life is concerned, is dead. Now, whether it had life before or not, that's a different story. But as far as life is concerned, it may be dead. But there's no a priori reason when you start doing the multiplicities in billions for what there are in the way of stars, galaxies, planets around them and all, it's going to be it. There's going to be one somewhere. Yes? If it came back negative, Yeah, they were. Well, this is a really good question, and I'm not doing that to be pandering, because it's negative doesn't mean there isn't anything there. We may be giving them totally the wrong food, and the only thing that might grow out, if it does grow out, would maybe be something we brought. So it's absolutely fantastic question. I asked a version of it on the graduate course that we taught for years. That you find you're nosing around on Mars and you find something that looks like a living system, some kind of a living cell. How would you prove that it was unique to Mars, or at least originated in Mars and didn't come from the Earth? And that's something we're going to have really good signatures, because some theoretical biologists say that life across the entire cosmos has probably driven to a 4-cohort base system and a peptide bond living system. That's what they say. And so if it isn't, then we'll probably have living things around us and we don't even know they're here. Okay? If it is like us, we'll probably be able to work our way through that. The food? This one, food? All right, well, got one. You know what? You probably haven't had your hand up for a while. Oh, I know who that is. How they do. Okay. We have an analog system he's referring to as a rotating bioreactor system. And it's about the size of a Campbell's soup can, but it's made of clear plastic. And if you fill it, the entire thing with water, all the way, or with fluid, with culture media, and you rotate it, an interesting physics phenomenon happens. It becomes actually, the fluid becomes coupled to the vessel, and the fluid turns with it. And if you put particles in it, cells that are a little bit more dense than water, then they will fall, and they fall continuously. So that's an analog. So they're in free fall for the entire time. They're at terminal velocity falling through the fluid for anywhere from hours to days to weeks to months that we've had them do this. And so that's what we were talking about in the analog system. Under those conditions, there are a lot of interesting things that happen. Cells get together. We don't know how and why. They do the same thing in space. They just eventually come together, and they start to then morph a piece of tissue that has many of the characteristics that the native tissue that the cells came from had. So, is that what you were talking about? Okay. I'm violating
enzymes, how do you explain that threshold statement?
How do I explain the fact that all these things would take place in an ordered fashion like this? How would it take place that cells get together, they do this, they secrete matrix, they actually become what looks like histochemically, it looks like a piece of tissue. And it's very, very, very difficult to explain, actually, as to why it would occur, because it doesn't occur when you put it in a flask or in a petri dish or in a tea flask. It doesn't occur as readily. And the reason, I think, is in part is that one of the things that happens in space is a freedom of co-location. Okay? Cells can come together and they stay together if they need to be together, and if they don't need to be together, they don't stay together. And the ones that need to be together come together. And if you put parenchymal cells in with mesenchymal cells, they don't segregate off entirely. They actually integrate with each other. And that's why we get all the differentiated effects that take place, is because they can do that. I'm getting about a third of what you said. How do I explain the streaming? Yeah. How does it happen? Oh, why do I think it happened? Now I'm out on a limb, okay. Who's got the saw? It's very, very hard mathematically to say that you could get the number of ordered mutations that are required. Just talking about radiation resistance. Mathematically, it's almost impossible to believe that simple mutagenesis and selection were involved, okay? And random selection. This is not a pressured selection, but random selection. I could understand it in the radiation environment. When you go out there, I think that makes perfect sense. But what's here on Earth, no. I think you wanted me to say they came in from some other planet, but not going to do it.